For mutagenesis of the protein, I am going to use Pymol. Pymol is a software for visualization of the biological molecules. Here we are going to open it. After opening, I am getting the protein by opening from the local hard drive, or I can even get it from the RCSP database. After writing the code for the protein, which is 4-letter code, and I am going to fetch it. This is the protein of kinase, in which 2 manganese atoms are represented by violet balls. Along with them, several water molecules are present. These are shown as red crosses. And the ATP molecule, which is normally present in the kinases, can be seen over here in the sticks. Now I am going to display the sequence. Here you can click on display and sequence so that you can see the sequence of the protein. These are the water molecules, which are represented by zeros, and manganese atoms. Here you can see them. ATP molecule, when we click on it, then it gets selected. These are the residues which are given in the 1-letter code, and even you can display the residue names, which are represented by 3-letter codes now. And we are interested in doing mutagenesis at somewhere in this protein. So I will take 26th number phenylalanine residue and will change it to alanine. So how we will do it? Let's see it. First of all, you will go to the wizard tab, mutagenesis, and then click on the protein. So after clicking on the protein, you will get some options on the right side of the window. Here we are presented by a message that we have to pick a residue for changing into another residue. So I will pick it, the phenylalanine, at 26th number position. Here you can see that a select a rotamer for this residue or pick a new residue have been given as an option for us. So we are going to take the option of no mutation and will click on it. Here all of the standard residues are given. Along with the standard residues, you can see some of the protonation states of the residues. I will discuss them later in another video, but at this time I am going to change the phenylalanine, which is at the 26th number position, to the alanine. So I will click on it. After clicking on it, I will apply it. So here you can see that 26th number residue is now alanine. You can do the mutagenesis or mutations at other positions as well. If you know about the mutations from some literature, or you are doing some kind of experimental work in which you have come through some mutations in the protein, then you can model them or change in the existing protein structure. So 26 number alanine is now here, and this can be represented by sticks. So we have done it. Now we are going to save it, file, export molecule. This is the option, and here we are going to take 1 ATP because 1 ATP is the structure, and without changing any other option, only look on thing that is it going to be saved in a single file. So it is going to be saved in a single file. I am going to save it. Here it is asking me about the PDB format, so I am taking the PDB as a format, and I am going to write the 1 ATP, which is the original PDB ID, along with the underscore and mutate it. And you can even write the position 26th phenylalanine to alanine. You can write this information so that know about it and you can do the work easily, and then save it somewhere on the local disk. So it is all about the mutagenesis. Now, after clearing all of these sessions here, we can even see the same file which have been saved earlier, and by doing the display and sequence again, we can see here that the 26 number is same alanine. If you want to check it in the Notepad++, you can open it in the Notepad++, and here are the numbers of the residues, this column 15, 16, and if we come to the 26 number position, this is the 26 number position which we have mutated earlier in the Pymol. So you can see here that the alanine is successfully incorporated against the phenylalanine after mutations. So that's all, guys, and if you like the video, please subscribe and like, and give me suggestions or your problems or queries so that I can make videos on those subjects as well. Thank you.